Hey guys, I'm back with Mendelian genetics part 2, and we're going to delve a little bit further than simply the simple dominance. Now, when we think about Mendel's experiments, he basically said that each gene has only two alleles, and one of those has to be completely dominant on the other. We know that that is not the case. That is not the way the real world actually works. So there has to be relationships beyond this. One such case is incomplete dominance. Incomplete dominance is basically when you have an appearance between the phenotypes of the two parents, such as in these carnations, you have a red flower and a white flower produce a pink flower. Now, if you notice, red is dominant, so big R would be red. Little r would be white. Now, the offspring, if you were to cross these two, the offspring would all be big R, little r, which makes them pink. Incomplete dominance is when the heterozygote individual actually shows the blended phenotype. So that's one type of inheritance pattern. Another type of inheritance pattern would be codominance. In codominance, you have two alleles that both affect the phenotype. You do not have a blending of the phenotypes, and you get both of them to appear. One such thing would be in blood types. In blood types, you have three alleles. You have A, B, and I, or which is O. Now, A blood, you can get it in two ways. Get a capital IA from one from both parents, you get A blood. Or capital IA, little I, get a recessive and a dominant, and you get A blood. B blood is the same way. Capital IB, or capital IB, little I. And then O blood is little I, you get both recessive traits. And then AB blood is whenever you get capital IA, capital IB. Now, to explain this, if you were to look on a blood cell that had O blood, there would be no antigens. A blood would only have A antigens on the outside, so it's only going to recognize things with A antigens. B blood would only have B's on the outside, so it's only going to recognize things with B. But AB blood is going to have both A and B antigens on the outside. Now, this explains why a person with AB blood can get blood from anybody. Because if they get blood from O, their body doesn't recognize it as being foreign because it doesn't have any antigens on the outside. If they get blood from a person with B, they have the B antigen, as you can see here, right? There's the B's, so they recognize the B's as being themselves, so they're good. If they get blood from a person who has A, they also can recognize the A as being themselves, so that they're okay. And of course, if they get AB. So a person with AB blood can get blood from everybody. A person with O can only get it from O, because if they get blood from anybody that has antigens of anything on it, then they see it as being foreign and attacked. But codominance would be when you have both of them show up. I'll give you another example. If horses were governed by codominance, this is totally false, and you had a black horse, and you were to cross that black horse with a white horse, and both of these were homozygous, the offspring would be a zebra. This is not true, but that's kind of a way to understand what codominance is. Another possible inheritance pattern is multiple alleles. Blood types is also multiple allelic. Not only is it codominance, but it's also multiple alleles. And it's because you have three alleles, right? The three alleles you have an A allele, you have a B allele, and you have an O allele. So you have two dominant and recessive, three different alleles. A lot of things can be governed this way. Blood types are just one. It's going to give you more possibilities. Now, pleiotropy is the next. This is when one gene affects more than one phenotype characteristic. Example is sickle cell anemia. Down here at the bottom, you can see the normal red blood cells, and then you can see blood cells that have sickle shape. They are actually changes the shape of the phenotype of the organism. All right. Epistasis is whenever one gene masks another, such as in mice. You might have this occur where you have two genes that are controlling a trait. So like the C, as you can see down here, the C allele or the C gene is controlling pigment. The B gene is controlling how much pigment there is. So if you have a big B and a big C, then you're going to be black colored mouse. If you have little b's and then you have a big C, you're going to be a brown color mouse. But then if you have two little C's, you're albino, no matter what B combination you have. So if you look over here in this cross, if you cross two heterozygote individuals, we would expect a 9:3 to 3:1 ratio, with 9 being black, 3 being a color, 3 other ones being a different color, maybe brown, and then 1 being albino. Well, actually what you get is a 9:3 to 4 ratio, with 9 being black, 3 being brown, and 4 being albino. So it eliminates one of the phenotypes because it ends up masking it. All right? And then the last one is polygenetic inheritance. This is when genes are controlled, traits are controlled by more than two or more genes. Well, easy for me to say. An example would be like skin color, height, weight, intelligence, behaviors. Anything we have a multitude are controlled by polygenetic. And poly means many, genetic means genes. Now, the last thing is human genetic disorders. And you just did some one-pagers on human genetic disorders. And human genetic disorders are going to be done by a pedigree. And just let me give you a few reminders. In a pedigree, the square represents a male, the circle represents a female. In this particular case, it shows widow's peak or non-widow's peak, or maybe attached or non-attached earlobes. Some disorders are caused by recessive alleles. That means you have to get them from both mom and dad to get it. Some are caused by dominant alleles. You have to get it from only one. An example of recessive would be cystic fibrosis. You have to get it both parents to get it. Tay-Sachs, sickle cell disease. Dominant ones would be Huntington's disease and achondroplasia, which is dwarfism, or it's called by dominant disease. But it's just kind of an introduction into those. Anyway, now the last thing is, how do we determine these human genetic disorders? One way we can do is testing. You can have amniocentesis, which actually would be able to test the organism before they are born. But no matter what kind of testing, you usually look at a karyotype. Remember what a karyotype is? A karyotype is a graphic representation or is a picture of the chromosomes. All right, so just remember what a karyotype is, and it can be used in diagnosis. All right, I hope this helps you a little bit to understand the last part of Mendelian genetics, and I hope you guys have a wonderful day.